All right, let's dive right into a truly fascinating paradox, a situation where a massive accumulation of genetic errors actually becomes a tumor's ultimate Achilles heel. Today in this explainer, we are breaking down microsatellite instability, or MSI. It's one of those rare topics that completely flips our traditional understanding of cancer biology on its head. Here is our roadmap for today. 1. DNA's copy machine problem. 2. The broken proofreaders. 3. How we test for MSI. 4. The immunotherapy plot twist. And 5. Beyond treatment Lynch syndrome.
Let's get started with part 1, DNA's copy machine problem.
So to really get this, we need to zoom way into the molecular level. Our cells act like incredibly busy but sometimes kind of clumsy copy machines. And to understand microsatellite instability, we first need to know what a microsatellite actually is. It's basically just a short, repeating genetic sequence. Think of it like a stutter in your DNA's code, repeating 1, 2, or maybe 4 letters over and over again. These little stutters are distributed everywhere across our genome, and because they are so highly repetitive, our cellular replication process really struggles to copy them perfectly every single time.
Now, what's really wild is how our cellular machinery handles those repeating letters. The actual enzyme responsible for copying your DNA polymerase, gets a little confused by all that repetition. It literally slips on these repeats. When it slips, it accidentally adds an extra letter or removes one from the genetic code, which creates what we call an insertion or deletion loop.
Moving right along to part 2, the broken proofreaders. Of course, our bodies usually have a built-in solution for these slipping mistakes. Normally, our cells have a fantastic built-in autocorrect, a mismatch repair system, or MMR, that immediately catches these typos and fixes them. But when that DMMR system is deficient or broken, those slipping mutations aren't caught. The genetic errors are literally allowed to run wild.
So, who exactly is doing all this proofreading? Well, think of these 4 specific proteins, MLH1, MSH2, MSH6, and PMS2, as your cellular proofreaders. They constantly patrol the DNA looking for those insertion or deletion loops. But the problem is, they can be disabled. Whether it's through inherited germline mutations, acquired somatic mutations, or epigenetic silencing, like MLH1 promoter hypermethylation, these proteins essentially get fired. And naturally, when they get fired, the typos stop getting fixed.
The crucial takeaway here is that this total lack of proofreading creates a hyper-mutable state. We are talking about a massive, overwhelming accumulation of frame-shift mutations across the entire genome. And that right there is the defining biological hallmark of microsatellite instability. It is pure molecular chaos. Which brings us to part 3, how we test for MSI. Let's step into the diagnostic lab for a second and see exactly how medical detectives spot these completely invisible typos. Basically, we rely on 3 main diagnostic weapons. They escalate from quick, basic protein checks all the way up to massive computational sequencing. We have immunohistochemistry, PCR-based analysis, and next-generation sequencing. Each of these has a very specific target and a unique advantage in the clinic. First up is IHC. Now, IHC doesn't actually measure the DNA stutter length directly. Instead, it just looks to see if the proofreaders even showed up to work. It detects whether MLH1, MSH2, MSH6, and PMS2 are physically present in the tumor tissue. Finding a missing proofreader protein, a loss of expression, means you have a deficient mismatch repair system, and that is highly concordant with high microsatellite instability. It's a fantastic, super fast first-line screen. Then we have the traditional gold standard, PCR. This takes a much more direct approach. It follows a specific 3-step molecular workflow to physically measure the length of the genetic stutters. You amplify the DNA from both the tumor and a sample of normal tissue, you run it through a sequencer, and you physically compare the lengths. If the tumor's DNA fragments are a totally different size than the normal tissue, bam, you've got instability. But researchers aren't just looking anywhere. Scientists check 5 specific markers, which are known as the revised Bethesda panel. If 2 or more of these specific mononucleotide repeats show altered sizes, the tumor is officially classified as MSI high. If only 1 is altered, it's MSI low. And if absolutely none are altered, the tumor is considered microsatellite stable, or MSS. Today, however, next-generation sequencing, or NGS, is absolutely taking over. And it's easy to see why. Instead of looking at just 5 little markers, it brings in advanced bioinformatics algorithms to scan dozens, sometimes thousands of loci all at once.
Plus, it frequently eliminates the need for a normal tissue sample for comparison. It just relies on established baseline data to give doctors a precise quantitative MSI score alongside a bunch of other critical tumor data. Okay, so we've mapped out the broken proofreaders, the molecular chaos, and all the lab tests, but honestly, why does this microscopic instability actually matter for a patient's survival? What does an MSI-H diagnosis mean when you're sitting in the clinic? And that leads us to part 4, the immunotherapy plot twist. This is the absolute coolest part of this explainer. This is where these mutations become a massive therapeutic advantage. The sheer volume of DNA typos we've been talking about creates a high load of really strange, weird-looking proteins called neoantigens. Because these proteins look so incredibly foreign, the tumor becomes highly immunogenic. The immune system's alarm bells are just ringing off the hook because it clearly sees this tissue does not belong.
Now, cancers are incredibly sneaky. They put up chemical shields to hide from the immune system. But because the MSI-H tumor is already heavily flagged, administering checkpoint inhibitors like pembrolizumab or nivolumab simply strips away the tumor's shield. This totally unleashes the body's natural defenses to devastating effect against the cancer.
This specific dynamic actually led to a true revolutionary milestone in modern medicine. The FDA approved anti-PD-1 therapy as a tissue-agnostic treatment. This is huge. It means the drug is approved not based on where the cancer started in the body, whether it's the colon, the stomach, or the uterus, but entirely on the specific genetic signature, MSI-H. If the tumor has the typos, the drug is cleared for use. And the plot twist goes even further for early-stage disease. Take stage 2 colorectal cancer, for instance. Knowing a patient's MSI status here does 2 massive things. First, it predicts a better natural overall prognosis. But second, it actually spares patients from undergoing harsh 5FU chemotherapy. Why? Because the data clearly shows MSI-H tumors in this stage just do not benefit from it. We literally save patients from totally unnecessary toxicity. And finally, part 5, beyond treatment, Lynch syndrome. Detecting these typos has implications that go way, way beyond the immediate treatment of an individual patient. Let's look beyond the tumor itself. When a pathology lab finds a deficient mismatch repair system, it immediately sets off a protocol alarm. It triggers what's called reflex testing, where doctors look for germline mutations in those MMR genes. Basically, they need to answer a big question. Was this genetic brokenness just a random fluke inside the tumor, or was the patient actually born with a broken proofreader gene? This reflex testing is a total game changer because it can diagnose Lynch syndrome, which is a hereditary cancer predisposition syndrome. If the mutation is inherited, it means the patient is at an extremely high risk for other cancers down the line. But even more importantly, it allows doctors to proactively screen, test, and protect the patient's entire extended family who might share that exact same broken gene. As we wrap up this explainer, it leaves us with an incredibly inspiring thought to ponder. If decoding a simple, clumsy cellular stutter can unlock universally targeted, tissue-agnostic therapies and actually protect entire families, what other microscopic secrets are just waiting to be found in our DNA? Thanks so much for joining me on this deep dive into microsatellite instability.